Hello, everybody, and welcome to another episode of Silence Abounds Science at Home, where we will discover home lab magic. I'm very excited by today's episode. We are going to explore and demonstrate the powerful molecular biology technique called Polymerase Chain Reaction, PCR. PCR polymerase chain reaction is a laboratory technique used to amplify DNA, meaning it makes millions of copies of a specific DNA sequence in a short amount of time. PCR takes a small amount of DNA and copies a specific region repeatedly, so there is enough DNA to study and analyze. So what is needed for PCR? First, you need a DNA template. The DNA template contains the target sequence. Next, you need primers, which are short DNA sequences that define the start and end of the region to be copied. You need an enzyme called DNA polymerase, which is a Taq polymerase, which is usually for DNA polymerase, we use Taq polymerase in PCR because it's heat-stable enzyme. Also, you need a supply of free nucleotides, A, T, C, and G, which are building blocks of DNA. Also, all that needs to be in a buffer which maintains optimal conditions. But the most important thing that we need in order to carry out a polymerase chain reaction is a thermal cycler. In this lab, we will use a thermal cycler, which is a laboratory instrument used to amplify DNA through a polymerase chain reaction process. It precisely controls temperature cycles to drive the biochemical steps that doubles DNA many times over. The thermal cycler uses a heated block for holding tubes and a programmable controller to switch temperatures precisely and rapidly. In this lab, we're going to be using PCR beads. These PCR beads contains everything we need to carry out PCR. Your enzyme, your free nucleotides, and buffer. And they come in these free dried beads. So let's begin today's experiment. And we're going to first prepare our samples ready for PCR. Okay, so now I'm going to prepare the PCR mix ready for PCR amplification. So I have a 0.2 ml PCR reaction tube here, and then I've got 4 labeled tubes, 1.5 ml tubes, and I've labeled them 0, 10, 20, 30, because what I'm going to do is pause at, I'm going to take an initial sample of the PCR mixture before amplification and put that into times 0, and then I'm going to pause after 10, 20, and 30 cycles and take a sample and put them each into the respective tube. Once I've collected all my PCR reaction solutions, they will then be run on Gel Electrophoresis to demonstrate the PCR reaction. So, first things first, So I'm going to add 20 microliters of primer mix, which I have here. Let me have a look. So I need primer mix. So I'm keeping my solutions on ice because they are very sensitive to temperature. You don't want to get them too hot. So I've got my primer mix here, and I want 20 microliters. So I'm going to get my 20 microliters pipette. Okay, so and I'm going to add that to my PCR tube. Oops. Let's hold the tube for now. Okay. I'm going to leave it in that one. And next, I'm going to add 20 microliters of ultra pure water, which I have here. 20 microliters of ultra pure water, and you want to use fresh pipette tips for each solution to avoid any form of contamination. I'm going to put that into my PCR tube. Okay. Next, I want to add 5 microliters of DNA primer. DNA template, I'm sorry. So I have my DNA template here. Okay, and we want 5 microliters. Okay. Okay, and then what I'm going to do is I'm going to give that a gentle mix. And I'm just going to flick it to mix it. Like so. Let's make sure it gets mixed. Okay, and then the last thing I need to do is add my PCR bead. And remember, the PCR bead contains everything, all I need for the PCR reaction, the nucleotides, the enzymes I need. So I'm just going to, and they come as a bead, so I just want one. There we go. Oops. Oh, I'll get that in a minute. So I'm going to add that bead very carefully to the reaction tube. And I want to try and get it into the solution. Like so. And it will dissolve, instantly dissolve. And again, you just want to gently mix it. And you should have an orange colored solution. You want everything to try and mix it as much as possible. And make sure your bead, your PCR bead, is fully dissolved. It is lyophilized, so as soon as it touches the liquid, it dissolves. It doesn't take much to dissolve, but I want to make sure everything is mixed well. If you have a vortex mixer, you can use a vortex mixer on a gentle mix, but I don't have that luxury. And what I'm going to do now is I'm going to put it in my centrifuge because I want all the reactant at the bottom of my tube. So I'm just going to pop it into the centrifuge briefly. And that should have put all the reaction chemicals to the bottom of my tube, which it has. As you can see, everything is at the bottom of the tube. So now we are ready for PCR. But first I'm going to take a sample of this. I'm going to take 10 microliters because this is time 0, because I want a sample where we've not had any PCR. So I'm just going to take 10 microliters and put that in my times 0 tube. So there's no PCR cycle. And then I'm going to add 15 microliters of ultra pure water. So and then I'm going to keep the tube on ice until I'm ready for electrophoresis. Okay, so this goes on ice. So now we are ready to set up and start the PCR reaction. Okay, so I've already pre-programmed my thermocycler. I got denaturation at 94 degrees for 15 seconds, annealing at 45 degrees for 15 seconds, and extension at 72 degrees for 30 seconds. And that's been set up for 30 cycles. So I'm now ready to load my cycler. I'm going to take my PCR tube that I have and put it in like so. I'm going to close the lid, make sure it snaps into place, and I'm going to click run. So there are 3 main stages of the polymerase chain reaction. The first one is denaturation and takes place between 94 and 95 degrees centigrade. The hydrogen bonds holding the 2 DNA strands together break, causing the double-stranded DNA to separate into single strands. Single-stranded DNA is needed so the primers can bind in the next step. The second stage is annealing, and this occurs between 50 and 65 degrees centigrade. And this is where short DNA sequences called primers attach or anneal to the complementary sequences on each DNA strand. Primers define the specific DNA region that will be amplified. And then the third stage is elongation, which usually occurs around 72 degrees centigrade, where Taq DNA polymerase, which is a heat-stable enzyme, adds free nucleotides to the primers, synthesizing new DNA strands. This step actually copies the DNA, producing new double-stranded molecules. Each strand becomes a template for the next cycle. So right now, this is the map that is going to follow, and it's going to do a cycle, and it's going to follow this for 30 cycles in total. So right now it's just preheating and getting ready. And once it's at the initial temperature, it will start the cycles. So the cycle has started. It is now on the second stage of the first cycle, and that's the annealing phase. And then it will go on after 15 seconds to the elongation phase at 72 degrees C. Okay, so we're going to let that run, and we're going to come back at cycle 10. We're going to pause it and take a sample of the PCR mixture. Okay, it's at the end of cycle 10, so I'm going to pause the PCR and then take a sample of the reaction mixture. Okay, so I'm going to open the lid, take my tube, and I'm going to pipette 10 microliters into my 10 microliter labeled tube. Like so. I'm going to put my PCR tube back in the PCR instrument and start the PCR again. And then to my 10 cycle tube, I'm going to add 15 microliters of pure, ultra pure water. And give that a little mix. And then again with the other tube, I'm going to leave that on ice until we're ready to do electrophoresis. Okay, so my reaction is now complete. It's completed 30 cycles, so I'm going to collect my final aliquot. Just like the other three, I need 10 microliters of reactant. Okay. So we're done with that, and now we're going to add 15 microliters of water. Give that a mix. Okay, so now I've collected all my samples, and we're ready for part 2, and let's see whether our PCR worked or not, and we will now perform electrophoresis. Okay, so I'm going to apply all of the PCR matrix. Okay, so I'm ready to pipette my samples into the wells, and I'm going to use the whole amount of PCR solution that I have left from the reaction, and that's what I'm going to pipette, about 25 microliters. And I've also got a DNA ladder solution that I'm going to use as a marker. So I'm going to pipette that first. Okay. And then I'm going to, I do not have any loading dye in my PCR products. So you're not going to see the bands, but they have an orange tint to them. So you should see some, you may be able to see something. So now I'm pipetting cycle 0, which is our baseline. And that does not have, that did not go through amplification. Next is the 10 cycle sample. Now the 20 cycle product. And I'm calling them a product because they are a product of PCR. And the last one, which is the 30 cycle sample. All right. So what I do now is I'm going to connect my electrodes. And then I'm going to run the electrophoresis for 30 minutes. All right, and I'm going to connect this to a power supply. Okay, this will run for half an hour. So here are our results. This is using a DNA stain to highlight the DNA products. And if you remember in lane 1, the first lane, we had the baseline sample, and that did not go through PCR amplification. As you can see, we cannot see any band in that lane, which is indicative that the DNA sample is so small that we are unable to detect it in our electrophoresis. The next lane had the sample that went through 10 cycles of DNA amplification. And again, we do not see a band. And again, this is indicative that we just don't have enough product to be able to detect it and visualize in our system. The next two lanes, we now start to see bands. So the next lane was our product after 20 cycles, and we clearly see a band. So we're starting to amplify our DNA sample, and we can now visualize and detect DNA. The last band, which is our product after 30 cycles, you can clearly see a band, and it is more intense than the previous band, which was 20. So we're making even more copies. We can see the intensification of the band is slightly more than the 20 cycle. And this is demonstrating an increase in copies being made. PCR has many types of applications and is used frequently in studying and detecting disease, and is used in disease diagnosis by detecting pathogens like COVID-19 and HIV. It's also used to study and diagnose genetic disorder screening and identifies mutations linked to diseases such as cystic fibrosis and sickle cell. And also, it is used widely in cancer detection and helps find tumor-specific DNA mutations and helps monitor cancer progression. It's used widely in forensics, in DNA fingerprinting, by identifying individuals from tiny biological samples like blood and hair. Check out a future video that I will be doing on DNA fingerprinting. Also, it's used in paternity testing by confirming biological relationships. It's used in agriculture and the food industry, in GMO detection, identifies genetically modified crops, and in food safety testing by detecting contamination by pathogens like Salmonella. Well, I hope you enjoyed that introduction to polymerase chain reaction. Please hit the subscribe button, it helps support my channel. Also, hit the like and share button. Feel free to leave me a comment. I love to hear your feedback, your opinions, questions. Send me an email. If you want to have an accompanying video notes that I also produce detailing the science behind the video experiment, please send me an email, which is in my video description linked, and send me an email requesting those video notes, and I'll be happy to send them to you for free. Until next time, keep on experimenting. Goodbye, everybody.